This is Biomed Cafe menu. Microfilament is presented today. We will talk about actin discovery, actin structure, actin function, and quiz game. I'm Dr. Biomed, cellular and molecular biologist. Welcome to the Biomed Cafe. In this cafe, complex biological and medical contents are presented in a simple, classified, and summarized form. Actin discovery. They saw something others didn't see. Actin was first discovered in rabbit skeletal muscle in the mid-1940s by Bruno Franz Straub. Almost 20 years later, H.E. Huxley demonstrated that actin is essential for muscle constriction. Cytoskeleton. The cytoskeleton is a network of filaments and tubules. It extends throughout the cell through the cytoplasm. It's located in all cells. It plays a role in giving shape to the cell, especially in cells without cell walls such as animal cells, in movement of cell and organelles, in organizing of cell, and in keeping the cell's organelles in their place. Cytoskeleton can be divided into three groups: microtubules, microfilaments, and intermediate filaments. Microfilament characteristics. They are also called actin filaments. They are the thinnest filaments in the cytoskeleton. Their diameter is 6 to 7 nanometers. They are flexible but also strong. Microfilaments are nucleated at the plasma membrane. Therefore, the edges of the cell generally contains the highest concentration of microfilaments. Microfilaments are quickly grow or assembling and shrink or disassembling. Microfilament structure. The major component of microfilament is globular actin or G-actin. 3 G-actin combine to form trimer actin, which continues to form filamentous actin or F-actin. 2 long strands of actin arranged in a spiral in order to form a microfilament or actin filament. Globular actin or G-actin. They bind to ATP for polymerization. Filamentous actin or F-actin. 2 long strands of F-actin arranged in a spiral. They are polar molecules. It means they have plus end or barbed end and minus end or pointed end. The plus end grows faster than the minus end. The terms pointed and barbed derive from their appearance under transmission electron microscopy. Microfilaments assembly can be divided into 3 phases: nucleation phase, elongation phase, and steady state phase. Nucleation phase. In this phase, 3 G-actin molecules slowly join to form a small trimer. During the nucleation phase, the formation of a stable actin nucleus occurs. Elongation phase. In this phase, the actin filament rapidly grows by the addition of many actin molecules to both ends. It can occur at both the plus and minus ends. Plus end grows relatively fast, but minus end grows relatively slow. As a result of ATP hydrolysis, filaments elongate approximately 10 times faster at their barbed ends than their pointed ends. Steady-state phase. In this phase, an equilibrium is reached as actin molecules join and leave the filament at the same rate, maintaining the filament's length. The filament length remains constant in the steady-state phase. The phenomenon is called treadmilling. It means new molecules are constantly being added to the plus end and falling off the minus end. A given actin molecule would appear to move along the strand. Whether a filament will grow or shrink and how quickly are determined by the concentration of G-actin around the filament. General structure of actin. Actin has two general structures: bundles and networks. Bundles. Actin bundles are closely packed into parallel arrays. There are two arrays types: polar arrays and nonpolar arrays. In polar arrays, all barbed ends point to the same end of the bundle. But in nonpolar arrays, the barbed ends point towards both ends. Networks. Actin filaments are loosely cross-linked and form 3D networks. Microfilaments isoforms. Microfilaments have three isoforms: alpha, beta, and gamma. Alpha is located in muscle cells: skeletal muscle, smooth muscle, and cardiac muscle. It plays a role in muscle contraction. Beta isoform is located in non-muscle cells. It plays a role in mediating of internal cell movement. Gamma is located in non-muscle cells too, and it plays a role in mediating of internal cell movement. Actin binding protein. Actin binding proteins are required for actin formation, continuance, and function. Function of actin binding proteins. They are involved in actin polymerization, depolymerization, stability, and organization. The dynamics of actin filaments are heavily influenced by various actin binding proteins. Actin binding proteins can be divided into 11 groups: actin nucleating proteins, G-actin binding proteins, F-actin regulatory proteins, filament cleaving proteins, branching proteins, capping proteins, cross-linking proteins, bundling proteins, membrane binding proteins, motor proteins, and cytoskeletal linker proteins. Actin nucleating proteins. They play a role in nucleation of new actin filaments. One example of actin nucleating proteins is formin. It binds to free actin ATP and brings them together. Another group of actin binding proteins is G-actin binding proteins. They play a role in nucleotide exchange, monomer sequester, and monomer delivery. One example of G-actin binding proteins is profilin. It promotes the exchange of ADP for ATP. Another example is thymosin beta-4. Thymosin beta-4 is a sequester of actin ATP monomers. And another example is WASP. WASP plays role in monomer delivery. F-actin regulatory proteins. They play a role in regulating F-actin. One example of F-actin regulatory proteins is tropomyosin. It strengthens actin networks. Another example is tropomyosin troponin. It regulates myosin 2 interaction with actin, filament cleaving proteins. They play a role in cleaving F-actin. One example of filament cleaving proteins is cofilin. It binds along 2-actin ADP in a filament, forcing a movement that destabilizes the filament and causes it to break. Another example is gelsolin. It inserts itself between actin molecules in filaments and disrupting the filament. Branching proteins. They play a role in branching actin. One example of branching proteins is ARP2/3 complex. It's a protein complex at the origin of branch F-actin networks by creating nucleation cores. Capping proteins. They play a role in preventing the addition or loss of actin monomers at end of the filament and stabilizing them. Examples of capping proteins are capsid, formin, and tensin. They bind to a filament's plus end. Tropomodulin and ARP2/3 complex. They bind to a filament's minus end. Cross-linking proteins. They play a role in dictating the formation of actin network structure. Some examples of cross-linking proteins are filamin, spectrin, and transgelin. Bundling proteins. They play a role in dictating the formation of actin bundle structure. Some examples of bundling proteins are vilin and fimberin. They bundle actin filaments in microvilli and fusin alpha-actinin. That they bundle actin filaments in filopodia. Membrane binding proteins. They bind actin to membrane. One example of membrane binding proteins is vinculin. It involved in anchoring F-actin to the membrane. Motor proteins. They play a role in muscle contraction. One example of motor proteins is myosin. It interacts with actin and generating muscle force. Cytoskeletal linker proteins. They play a role in linking actin to other cytoskeleton. Some examples of cytoskeletal linker proteins are spectrin. It links actin to intermediate filaments. Plectin and MAP2. They link actin to intermediate filaments and microtubules. And tau. It links actin to microtubules. Microfilament functions. Microfilaments have many functions. 1. Cell movement, involving in amoeboid movements of certain types of cell, developing of various cell surface projection including filopodia, lamellipodia, and stereocilia, helping to generate the forces used in cellular contraction and basic cell movement in association with myosin, and involving in some movement such as the crawling of a white blood cell in immune system. 2. Cell division. Microfilaments aid the process of cytokinesis, which is when the cell pinches off and physically separates into two daughter cells. 3. Cytoplasmic streaming. Cytoplasmic streaming is the flow of cytoplasm, the contents of the cell, including the fluid part called cytosol and cell organelles, throughout the cell. Cytoplasmic streaming transports nutrients, waste products, and cell organelles. 4. Intracellular transport. Microfilaments serve as highways inside the cell for the transport of a motor proteins called myosin, including protein containing vesicle and organelles. 5. Maintaining the structure of the cell. Microfilaments, as a part of the cytoskeleton, keep organelles in place within the cell and provide cell rigidity and shape. Muscle contraction. In muscle cells, actin works together with the protein myosin to allow the muscles to contract and relax. Cell signaling. The actin cytoskeleton also facilitates the transduction of mechanical signals. 8. Formation and maintenance of the cell junctions. One of junctions components is actin that forms and maintains junctions. 9. Cellular adhesion. Actin involves in cellular adhesion, especially in epithelial cells. Macropinocytosis. In macropinocytosis, actin filament polymerize under the cell membrane and it pushes the membrane forward into ruffles. Apoptosis. During apoptosis, enzymes degrade actin into two fragments. Thank you for watching. Please subscribe to this channel, and if you like the contents of this video, please like it. Now it's time for the quiz game. I have put the link to the quiz game in the description of this video. Download it for free and enjoy it. Until the next video, bye.